lambda EMBL3A for the construction of genomic library. Now you have the idea that different lambda vectors can be used for cloning our gene of interest, and one of the vector is lambda EMBL3A. So if we compare it with previous strategy of Maniatus, where he used two restriction endonucleases to generate DNA fragments that have blunt ends, because in Maniatus strategy enzyme He3 or ALU1 have been used that produced blunt ends, and they can recognize tetranucleotide sequence. Whereas when we are going to use lambda EMBL3A, so this strategy, it is comparatively more simplified. The reason is that here the source DNA, it is not digested with two different restriction endonucleases, rather it is digested with a single restriction endonuclease like SA3A. तो इसका एडवांटेज क्या है इस रिस्ट्रिक्शन एंजाइम को यूज़ करने का? तो इसका एडवांटेज ये है कि जब हम डीएनए को इससे ट्रीट करते हैं तो इट में प्रोड्यूस कोहेसिव एंड्स, और कोहेसिव एंड जो है वो आपस में लाइगेट कर सकते हैं. तो डिफरेंट जो डीएनए के फ्रेगमेंट्स हैं वो लाइगेट करके तो लार्जर डीएनए के फ्रेगमेंट्स बना देते हैं. तो SA3A1 से ट्रीट करने के बाद हम डीएनए फ्रेगमेंट्स को एल्कलाइन फॉस्फेटेज से भी ट्रीट करते हैं ताकि इसके 5 प्राइम फॉस्फेट ग्रुप्स जो हैं वो रिमूव हो जाएं ताकि ये आपस में सेल्फ लाइगेट ना कर सकें. तो उसके बाद हम जो वेक्टर डीएनए है, एंड हियर इट इज़ लैम्डा EMBL3A, इसको डाइजेस्ट करते हैं BAMH1 से और EcoR1 से. BAMH1 से डाइजेस्ट करने का एडवांटेज ये है कि ये सेम कोहेसिव एंड प्रोड्यूस करता है जैसे SA3A है, तो इस तरह से इनकी लाइगेशन इजी हो जाती है. और बाद में EcoR1 भी हम यूज़ कर सकते हैं ताकि जो नॉन एसेंशियल डीएनए है या फिलर डीएनए है उसको हम रिमूव कर सकें. और बाद में इसको बिकॉज ऑफ द प्रेजेंस ऑफ suitable selectable marker the recombinants can be selected. In next diagram we will explain that how lambda EMBL3A can be used for the construction of genomic DNA libraries. So first it is the source or the genomic DNA, and it can be digested with suitable restriction enzyme, and here it is SA3A, and later on it is treated with phosphatase so that 5 prime phosphate ends they are removed so that they will not ligate together. And then on the other hand, lambda EMBL3A, it is showing the genetic map. It contains different polylinker sites that contain restriction sites for different endonucleases like BAMH1, SOL1, EcoR1, and within the central non-essential regions the genes are present that are called as RED plus and GAM plus. So these genes are required for the replication of the lambda genome. When these genes are present, it will not be able to infect a suitable cloning host, which is P2 lysogen of E. coli. Infect P2 lysogen of E. coli, so no plaques are formed. That will indicate that foreign DNA it has not been ligated. Whereas if this region it is deleted, then lambda EMBL3A, it will be able to infect P2 lysogen strain of E. coli, and it will produce plaques that will indicate the insertion of foreign DNA at this location within the non-essential gene. So first it is digested with BAMH1 and EcoR1. So what will happen in the next step? So discrete fragments of lambda EMBL3A will be produced having BAMH1 cohesive ends here, and then the non-essential region like RED plus and GAM plus, they will be removed by EcoR1 digestion, And polylinker sites here at the left arm and the right arm, they are discarded after precipitation with isopropanol in the next step, and then they are discarded. Now we get left and right arm of the vector DNA, and here it is ligated with target DNA that contain our gene of interest. So they are mixed together and ligation will be there in the subsequent step. So in the next step, the foreign DNA, it is ligated here, and then it is used for in vitro packaging in the next step. So phage particles are produced, and then the recombinants, they are selected in the absence of sp I negative strains. Here in the next step, sp I negative indicates that red and gam regions, they are absent, so that only the phage particles that contain recombinant DNA, they will be able to produce plaques on P2 lysogen. So it will act as a selectable marker.